You might get an opportunity today that some people that didn't show up might get. One of those opportunities is this colloquium. If you're interested in science teaching, the biology department seminar today at 3 is on careers in science teaching. So if you're interested in getting information from people who've gone through the program and are science teachers in local schools, please come check that out. So we're into the sex chromosome topic now, but quick review on meiosis, because everything that we do from here through the rest of the semester depends on meiosis. The transmission of chromosomes from parents to kids. We're going to talk about this when we talk about transmission genetics. We're going to do pedigree analysis later in the semester. It all depends on understanding how these chromosomes and their mutations flow from parents to kids. So another worksheet to work on in class. Don't start it yet. It's a brief explanation. This should look a little bit familiar. We're diploid individuals. We've got 2 pairs of every chromosome. This is a cell that I've diagrammed that is a diploid cell, except it's just gone through synthesis, post-synthesis. In other words, there should be how many copies of every double helix? 2 copies of every double helix. We call those copies, like those 2, that are genetically identical to each other. They have the same alleles of the same genes, capital Fs and capital Gs. That thing that's circled now in blue, we refer to as what? That combination of alleles, 1 or more, that are on the same piece of DNA. That's a haplotype. So there are 2 different haplotypes in this cell of chromosome 13. There's capital F with capital G, right? And that one's been synthesized. There are 2 versions of that. There's a capital F and a capital G there, and its copies over here. And there are 2 copies of the other haplotype, capital F with the little g. Likewise, we've got 2 copies of capital H on chromosome 22 and the other allele in this individual of H, lowercase h. So what was the genotype of this cell before synthesis? There will be 2 copies of chromosome 13. The 2 homologs, 1 from mom, 1 from dad, the paternal version and the maternal version, will be 2 chromosome 22s. So what were their haplotypes? Chromosome 13. If we work backwards from DNA synthesis, what happens? You go from 4 chromosomes, 4 sister chromatids here, to 2. So what are their haplotypes? What goes on each? What are the genes? What are the versions, the alleles on each of those pieces of DNA? FG. So is it capital F or capital G, capital lowercase f, lowercase g?
Capital F.
Why do you sound so sure that it's a capital F? There's a good reason. Right, so if after synthesis, all of the chromosomes only have 1 allele of gene F, the uppercase version, then, damn right, there are only capital Fs in the cell before synthesis, because you can only make copies of what was in the cell before synthesis starts. So then how are the Gs arranged relative to the Fs? If there are 2 different alleles, 2 different haplotypes, a capital F with a capital G, and there are 2 of those, that means one of those is the original and one's the copy. And if there are 2 capital Fs with lowercase g's after synthesis, it means there had to be a lowercase g with a capital F before synthesis, which means that one of those capital Fs is with a capital G and the other one was with lowercase. So you've got 2 copies of every chromosome after synthesis. You work backwards, basically just cross out. Another way to do this would be to cross out one of the duplicates of each type in the post-synthesis cell And that gives you what the genotype was pre-synthesis. So we would call that capital F over capital F and capital G over lowercase g if we were writing the genotypes independently, or if we were using haplotypes, it would be capital F capital G over capital F lowercase g. So that's pre-synthesis, post-synthesis. I'm going to erase some scribbling now. Okay. So if that's the complement of DNA that's in the cell post-synthesis, this should look kind of familiar to one of the questions that was on the exam. What's not showing up here is the dotted line that shows where this chromosome, or this cell is about to divide. Is this arrangement of chromosomes correct or incorrect? Down here. There's something really important about we're about to do the first division of meiosis. There's something really important about that two chromatids stay together at the first division, called which chromatids? The sister chromatids. Two versions that are exactly identical to each other. Are those next to each other on one pair on each side of the cleavage furrow? Are the two identical haplotypes next to each other? And are they each on one side or the other side of where the cell's going to divide? Yeah. Is this the only way you can arrange these chromosomes at metaphase? What would be the other way you could arrange these chromosomes, and there would still be something that would happen in biology? Switch the sides of
capital G, lowercase g.
Right. So you could take that pair of sister chromatids and that pair of sister chromatids and swap places. In other words, you could have a cell that was the two capital H's on one side, lowercase, on the left side, capital H on the right side, and instead you could have the capital F with the lowercase g on the left side, and the capital F with the capital G on the right side. This is important for meiosis, which is why I'm repeating this, because when we start talking about sex chromosomes, this is going to be really important. There are two different ways to arrange these chromosomes at metaphase. Okay, so the question now is, and work on this by yourself, or talk to a neighbor. Part A up there in the upper left-hand corner. So we've gone through the setup of this problem now. That we've got this cell that has the chromosomes as they're arranged right before meiosis starts. So how are those chromosomes going to segregate? There are three different pathways here. One way to do the first division, the second way to do this first division, and a third way. So that would result in either those two cells, path 1, after the first division, or those two cells after meiosis 1, division 1, second choice, or here's your third choice. So the first question in this upper left-hand box, we've got a minute or two to do this. Which of those three arrangements, 1, 2, or 3, is possible biologically? You can choose 1, 2, 3, or any combination of them. There might be one right answer, there might be no right answers, there might be multiple right answers. So is it pathway 1? If this post-replication cell divides, will it produce those two cells?
Yes.
I heard one yes. I need more.
Yes.
Yes? Okay. Not just more yes. Does anybody think no? Why is it yes? How do you know it's yes? What are you looking for to say that it's yes?
After meiosis division.
Right? So if the cell is going to divide and make a cell with those chromosomes and those chromosomes, which is what that dashed line represents, then do you get exactly the same genotypes in those resulting cells? Yes. Okay. What about pathway 2? And what about pathway 3?
In pathway 2, they switched around.
Okay, so in pathway 2, for example, the capital F with the lowercase g should be with uppercase H, but down here, the capital F and lowercase g are with lowercase h, right? If the cell is going to divide in between those pairs of sister chromatids, you won't later find that the other has occurred. Now, I want to reconcile this with, so it's definitely not pathway 2, but it is true that this is another arrangement of the way that the sister chromatids could arrange themselves at metaphase 1. We just talked about that about 5 minutes ago. So this could happen. This will happen in meiosis. It just won't happen from this starting cell. It would happen from the cells as I drew them to its right. You'd get those combinations of haplotypes in those cells. So as it's drawn, this pathway wouldn't occur, but it can occur in other meioses resulting from other cells. What about pathway 3?
No.
Capital F, lowercase g should be with capital F, lowercase g is with uppercase H.
It's the last part that's all messed up.
Okay, so wait, what do you mean by the last part?
The gametes.
Yes.
Oh, so the second division. We're not at the second division yet. So does the first division work properly?
Yes.
The chromosomes all go into the cells they're supposed to go into? Okay, so the answer there is 1 and 3 then. That one's okay. But now the question in the upper right-hand corner is what happens when you do the second division? Which of the different sets of 4 gametes that are shown on the outside of this chart are accurate and which are not accurate? So another couple minutes to think about that if you haven't already.
So,
Yeah.
I don't think I understand.
Assuming that all of the arrangements, including this one, are accurate, if those cells went through the second division, are the gametes represented appropriately for all 3 different sets of the metaphase 2 cells? Where's an issue? So what's wrong with 3? This one, the lowercase Roman numeral 3.
The second copies of the chromosomes on there. Some disappear.
Okay, so we've got, these gametes have chromosome, whatever number I called that chromosome, 23, I think, or 22 or something. They've got gene H, but not, what are they missing? Gametes should have 1 copy of every chromosome and 1 copy of every gene. So somehow these gametes lost the chromosome that has F and G, and these ones lost the chromosome that have H. So that is not how the second division would work. What about pathways Roman numeral 1 on the left and Roman numeral 2 on the bottom? Are there any unexpected haplotypes? Or are those all okay? we've got capital F, capital G, lowercase h. Those show up in two gametes. Capital F, capital G, lowercase h, or uppercase h show up there from that cell. So that's good. So pathway 1 and pathway 2 are accurate. So what is it about the gametes, just to refresh, what is it about the gametes in the pathway on the right, pathway lowercase Roman numeral 3? What's wrong with those gametes? We said it once, say it again. What's wrong with, what's incorrect about these gametes?
Or all the copies of the chromosomes.
In other words, so they're missing copies of chromosomes. How many copies of every gene or chromosome should every gamete have? 1. Diploid organism produces gametes that have half as many chromosomes. Diploids make haploid chromosomes. So those sets in 1 and 2 are haploid. They have 1 copy of every chromosome, and they're aneuploid. They're missing some chromosomes in the pathway on the right. One other really important comment about this, again, not important because I say it's important, but important because everything else we're going to do in class builds on it, is, are these gametes genetically identical to each other? Say in the pathways that led to actual genotypes or haplotypes of gametes, pathways 1 and 2, on the left here and on the bottom. For example, far left, is this gamete identical to this gamete, or the two that are on the far left? Are they genetically identical? Why or why not? You could argue, I think you could argue both. What's similar about them? What's similar? Those two gametes. What's different? Yeah.
One of them is homozygous dominant, whereas the other one is heterozygous.
Okay, so one of them has, some of these gametes have a capital F and a capital G, and the other one has capital F and lowercase g. Not zygosity, those are haplotypes, but that's okay. So that's the difference between them. There's a capital G in one and a lowercase g in the other. What's similar about these two gametes? Do they both have gene F? Discuss. Do both of those gametes have the same gene? Do they both have gene F? Is there an F on the screen? Yeah, right. So what's different about the Fs? The Fs are the same allele of the same gene, capital F, different alleles at G. One version of the gene in one gamete, a different version in a different gamete. And we've talked briefly about the fact that alleles mean that there are genetic differences between those genes, which is why we call them different versions or alleles. The sequence, that is, if we zoomed into those chromosomes, the sequence of gene G would be different subtly, maybe, than the sequence of gene lowercase g. But they do have the same gene. It's there, it's just a different version. So then back to what I'm hoping will turn into a discussion. I know it's Friday, but help me out, if you can, or if you're willing. Are the genotypes, or sorry, are the gametes genetically identical to each other? No, because they have different alleles at G. So this gamete is not genetically identical to that one. If they were both fertilized by genetically identical sperm, they would produce two genetically different individuals. Those two gametes are not genetically identical. Are there gametes up here that are genetically identical in the tetrad? Which ones, for example? The top two. The results of the second division from one of these, right? The result of the first division, those produce two genetically identical gametes. Because if you think about the genotype of this cell after the first division, it's homozygous. Capital F over capital F, capital G over capital G, lowercase h over lowercase h. So if you total up, question C, finally, to end, then we'll move on to something more interesting and a little bit more engaging, hopefully. One can hope. How many different gamete haplotypes are shown on this figure? In all of the gametes that are produced by, that are produced by accurate processes, the ones on the left and the ones on the bottom, how many different haplotypes are there? So what are the four? I heard four, we'll go with four, but the important thing is, what are the four haplotypes? If you write them out. Okay, so capital F, capital G, and then what? And then a parenthesis, or a semicolon, H. Which did that come from? That came from those gametes. Okay, what are other haplotypes? Capital F, lowercase g, lowercase h. Where did those come from? Okay, those ones. So those two are the top haplotype. Those two are the second haplotype. Those ones are capital F, capital G, lowercase h. And the last haplotype, capital F with lowercase g and uppercase h. So in meiosis, we started from the same cell up here at the top. Right, one cell. And we produce four different types of gametes. They're genetically similar because they have the same chromosomes and the same genes, but they're genetically different. There are four different, genetically different types because of different combinations of alleles. So let's talk about sex. This is not supposed to be the engaging part, by the way, but let's talk about sex. It's not permitted. What are the sex chromosomes that humans have? What do we call them? X and Y. So if I was going to do male meiosis, what would be the sex chromosomes found in the cell that starts meiosis? What are the sex chromosomes that males have? X, Y. So if we wanted to draw out male meiosis, and we ignored all the autosomes, chromosomes 1 through 22, we just looked at what the sex chromosomes were doing, then we'd have to do synthesis first. So we'd have two X's and two Y's. This is maybe the most easy time in the world to see sister chromatids. Which are the sister chromatids drawn up here in the post-replication cell? The X's are replicates of each other. They're copies of each other. So those are sister chromatids. And the Y's are sister chromatids. So what do these look like after the first division? How do those chromosomes segregate, assort into the two resulting cells? Sister chromatids stay together at the first division. So right now, what sort of gametes are we making? What type of gamete is going to be produced? Sperm. It's male meiosis. So at the next division, division 2, we're going to make 4 spermatids. So what are the haplotypes of those 4 cells? That is, what chromosome do they have in them? X, X, Y, Y. What happens when females do meiosis? What's the sex chromosome constitution of a female diploid cell?
2x.
It's XX. And by the way, we're going to talk about this a lot more next class, but when I say male and female, because this is a genetics class, I'm talking about chromosomally genetically male and female. We're not talking about gender, we're talking about sex. We're not talking about perception, orientation, we're just talking about from the chromosomal basis, are these individuals male or female? So males have an X chromosome and a Y chromosome, females have two X chromosomes. So after replication, there are four X chromosomes. After the first cell division, there are two XX cells. Oh, but let's see. After the first cell division, I probably should do something a little bit differerly, right? How does female meiosis differ from male meiosis? Right, so I should draw one that is going to be the oocyte, and one that's the first polar body. So the bigger one being the oocyte eventually. After the second division, the primary polar body, first polar body divides, and the oocyte has another asymmetrical division to produce the oocyte. You probably already know this. I don't know if you know this. What is the sex chromosome haplotype of every oocyte a female produces?
X.
Women always put an X chromosome in their oocyte because that's the only sex chromosome you've got. So what I think you probably already know, and you maybe know why, is who controls the sex of the kid? We don't really control it. It's random. But what are the ratio of X-bearing sperm to Y-bearing sperm? That is, if one of those sperm you chose at random and fertilized the X-bearing oocyte with it, what's the chance that you pick a sperm that's got an X chromosome?
1 out of 2.
1 out of 2, 50%. There are two, every time a male produces sperm, half of the sperm carry an X and half carry a Y. Whether you're looking at one meiosis like we did here or every meiosis. Every time meiosis happens, you produce 4 sperm, 2 carry an X, 2 carry a Y. So the probability of an X-bearing sperm fertilizing that necessarily X-bearing oocyte is 50%. The other 50% of the time, a Y-carrying sperm will fertilize the X. So when an X-bearing sperm meets an X-bearing oocyte, you get an XX individual, which is a female, and the other 50% of the time, you get a Y from dad, and again the X from the mom's oocyte, and that's a genetically male organism. So that's why human sex chromosomes or sex ratios rather, are about equal. About half males on the planet and about half females on the planet. Although there are interesting reasons why it's actually female biased, but we don't have time to talk about that right now, unfortunately. There are more females on the planet than males, but by a small margin. So at this point, please don't collaborate this time with each other, but I would appreciate it if you would answer a few questions on Socrative, and we'll move on from there when we're done. So let's go back to this for a second. Okay. So does the picture on the left look like the picture on the right? Yes or no? Shake your head or nod your head. Don't even have to say anything, don't have to raise your hand. Pictures are same or different? They're different. So how many sister chromatids are circled in blue? 2. Each of those objects, each of those things is a double helix. So how many total sister chromatids are on the left side? There are 46 things that we colloquially refer to as chromosomes, but each one of those things, like the one that's circled in blue in the upper left-hand corner, is 2 sister chromatids. So what's the product of that? 92. Okay. Same on the right side. 92. Why is that? Some of you got that, some of you didn't. That thing is that thing. We talked about it in class a couple times. Different stages of metaphase. One stage of metaphase on the right, early metaphase, where the arms of the 2 sister chromatids are still glued together, so you can't see that they're separate. But every object that we see at metaphase is a pair of sister chromatids. So both of those blue circled objects are 2 sister chromatids next to each other. They're either spread apart or they're glued together. Same amount of DNA in both of those blue circles. So it's also 92 on that side. So I'm hearing a lot of conversation, which is good. I appreciate that. Does anybody have any questions? It's not at all obvious. It's true, but it's not obvious. So if I can do anything to make it more obvious or more clear, I'd love that opportunity, if not now, in office hours or some other time. By the way, these are both human karyograms. So all of these images are at what stage of the cell cycle? They're all metaphase. They're both karyotype 46. One's XX, one's 46XX, the one on the left. The one on the right's 46. Oh, they're both XX. Huh. Silly me. Look, 2 X chromosomes in each. So they're basically genetically identical pictures from 2 different individuals. Different stages of the cell cycle. So we talked about the fact that this is false because different gametes will have different haplotypes. But what's the ploidy of every gamete? How many copies of every chromosome should it have? Everyone got this question right, so you should be confident in your answer. How many copies of every gene or every chromosome in a gamete? 1. Or chromosome and chromosome. So haploid. And the reason I know you all knew this is because almost all of the answers up there were a combination of A and C in the Socrative quiz. So I think everybody, or almost everybody at least, realized that haploid means both one homolog, one chromosome, and also one copy of every gene. Is you should not have 2 copies of a chromosome or 2 copies of a gene in a gamete, which is why B and D are not correct. The reason that we just spent a class period doing that may be obvious or may not be, but these were concepts that were in the half of the quizzes or exams I've graded so far frequently missed. If after the short introductory re-lecture I gave at the start of class, you got the concepts right on the Socrative quiz, you will get points for those concepts on the exam if you missed them. In other words, if you got them wrong in the exam and you got them right today, after I explain the concepts again, then you got points for it. On the exam, not just quiz points, but exam points. If you got them missed again today, please come see me. You'll see after you get your quiz for this, the grade for the Socrative quiz today and the grade for your exam next week. I would like to talk to you one-on-one because I want to help, if I can. Any questions? More conversation. Questions? I just retested the 2 most commonly missed questions from last week, in other words. Because maybe I phrased the questions on the exam strangely. Just checking. For next week, we're going to pick up this chi-square analysis spreadsheet. So Monday, that's what we're going to work on. I will push back the deadline on Google Classroom again until Monday, so not due tonight now, due next class after we get some time to work on that some more together in class. And where we're heading with this, because this is all very abstract, we're doing spreadsheets, chi-squares, flipping these poker chips that I brought again just in case, has entirely to do with sex chromosomes and the gender of kids. Because how many different genetic sexes do humans have? 2. We've got genetically male and genetically female. We've got XX and XY. And we just talked about the fact that dad's sperm basically make the decision. Does the sperm have an X in it? That creates a female, genetic female. Or if the sperm's carrying a Y, it produces a genetic male. And we've also discussed, although briefly, our expectation. That if meiosis works as we predict, half of the offspring should get an X from dad and be female, and the other half should be male. That's basically a coin flip. If one man, and we're going to talk a little bit more about this next class, if one man had an infinite number of kids, if he was really promiscuous, then half of his kids should be female and half should be male. It's a coin flip, just like the exercise we did with the poker chips last time we met. Not the exam, but with the time before. Because this is a model for sex determination. Flipping a coin, flipping a poker chip. Half the time it should come up male, half the time it should come up female, if we're considering that this is the probability that a sperm carries an X or Y chromosome. And we can use that to do statistical analysis of data sets to find out if you really are getting males that half of the time send an X chromosome to their kids and half the time a Y. So this is just a device to mimic things that actually happen in nature, like the choice of the random, presumably, choice of X or Y chromosome in this sperm. So next time we come and we finish up this spreadsheet on the poker chip flipping, we're going to move on to analyzing data sets from humans about sex ratios.